Hello, my name is Martina Delgado and I'm going to present a work which is the result of a collaboration between researchers of the Polytechnical University of Valencia and the University of Valencia. It is the development of a photonic biosensor which is based on the interaction of the evanescent optical field in a microfiber with a periodic network of molecules fixed on its surface. We call these devices biogratings. The fabrication process of these BBGs comprises different steps. First, we have to fabricate the microfibers that have a diameter of a few microns. Second, we will attach a periodic network of, in our case, BSA proteins to the surface of the microfiber by microcontact printing, and this will constitute the incipient optical grating. When we immerse this device in a solution that contains the specific target that will bind to this bioreceptor, the modulation depth will be increased as they attach one to each other. Here I'm showing a FISM image of one of our devices, which was fabricated in a taper of 5 microns. When we illuminate our devices with a broadband light source, the evanescent optical field of the mode guided by the microfiber interacts with the bioreceptors, and it will appear a reflected peak located at the Bragg wavelength corresponding to the period of this network of molecules. Here I'm presenting a collection of spectra that were recorded during the fabrication process of a device. We used a fiber Bragg rating, a conventional fiber Bragg rating, in order to monitor the loss that we could be introducing during all the fabrication process, and that they turned out to be very small. So this is the case of a single microfiber with not any molecule on it, so there is not any other reflection peak than that of the FBG. But when we introduce the network of BSA molecules, it appears a weak reflection peak located at the period determined by the structure. As the targets bind to the bioreceptor, this reflectivity of the peak increases, and then one could quantify the concentration of targets in the solution by means of measuring the variation of this reflectivity. There are different parameters that we can optimize in order to improve our biosensor. The first one is the diameter of the microfiber, and here we present the calculated reflectivity of the BBG as a function of the fiber diameter. We observe that below 5 microns we can have significant gratings, and here we compare experimental data, which are the points, with the theoretical curves that are the solid lines. This curve corresponds to the BBG formed by BSA plus the targets, and here these are only the BSA molecules, and theory and experiment agree quite well. Finally, we decided to work with tapers of 3 microns as a compromise between the reflectivity of the BBG and the fragility of the samples. One advantage of our proposal is that we can multiplex different BBGs in a single device. Microcontact printing consists on placing in contact a stamp where we have already the network of BSA molecules with our microfiber. Then, by rotating the stamp with respect to the axis of the microfiber, we can change the period in an easy way. Here we see how, by rotating this stamp in just 20 degrees, we tune the Bragg wavelength in more than 100 nanometers. So we fabricated, as a proof of concept, a single device with a microfiber of 5 centimeters long that had 2 BBGs printed on it of 2 different periods. And the result was to observe 2 different reflection peaks that were well resolved in the spectrum. We characterized the performance of our biosensor and did a dose response immunoassay, and the experimental point fitted very well to a sigmoidal regression, which is the conventional curve that one would expect for these experiments. From these results, we quantified the limit of detection and limit of quantification and obtained these numbers, which are level-free conditions, and these numbers are compatible with many other results in the literature, so this is a quite promising result for a first proof of concept. As a conclusion, we have presented a photonic biosensor that combines the sensing capacity of a microfiber with our ability to pattern periodic networks of molecules on its surface. We characterized the performance of this device as a biosensor and obtained promising results from the limit of detection and quantification, and we also demonstrated the capacity of these devices to be multiplexer. Thank you very much for your attention, and I hope that you find this work interesting.